So we go through, here's the link. Also attached to this for this PowerPoint, I have just a couple of slides because maybe some of you who aren't really familiar with epigenetics, you're wondering, and we've had questions about it. Remember, I think we talked about, somebody was asking, and then Elizabeth answered for us the methylation, how methylation is working. We're still not completely, we don't understand the complete dynamics of why the acetylation of the histones do what they do, or why the methylation does what it does. We kind of think that with the acetylation, it's kind of opening up space so that the genes can be turned on. The methylation is maybe getting in the way of the cellular machinery so that it doesn't get expressed. But here are a couple of slides. First, what are any questions or comments on the video? When I thought it's kind of an old video. I didn't realize it was as old as it was. I remember kind of when it came out, but you couldn't access it online until I think fairly recently. So it came out maybe 5 years ago, or maybe even a little bit more. So that's the other reason I wanted to post that epigenetics paper, is because it's from 2012. So it has even a little more updated stuff. Comments, questions on epigenetics? Okay, everybody understand what's going on? So imprinting, everybody understand what imprinting is? So imprinting is basically the sort of memory of the cell that you came from. So whether that chromosome, or from the chromosome that the gene is located on, so if that chromosome came from the father or came from the mother, the offspring are kind of remembering which one. And so with those conditions that they were talking about originally, depending on which chromosome was inherited from your father or your mother, you had a very different sort of phenotypic condition, right? And they were trying to explain that. That's explained through imprinting. Basically what the imprinting is on these different chromosomes. And if you think about it, not all of our genes are the same. And so we can have 2 copies of a gene, say, on a chromosome in some sort of cell, and whatever that gene is, we're just fine having 2 copies and cranking out to, having those 2 copies working functionally. But then maybe somewhere else on that chromosome, whatever that gene that's being produced, maybe the cell or the organism is sensitive to the amount that is being produced. And so we have to silence, or we have to shut down, turn off one of those genes. We do that through that DNA methylation, and that's what happens with the imprinting. So I'll show you here a slide. This next slide, it'll show you the specific example from the video. Acetylation of the histones is basically increasing the gene expression. The methylation on the cysteine nucleotide is basically associated with transcriptional repression. Okay, so that's how you're that's kind of how that gene regulation is happening. Okay, so in the video, you had the 2 different conditions, right? The Prader-Willi and then the Angelman, right? Here are the chromosomes. So here's your paternally inherited chromosome. What is it, chromosome 15, they said? I can't remember, I think it's 15. Here's your paternally inherited chromosome. This is your maternally inherited chromosome, right? So on the paternally one, it has an active Prader-Willi gene and an imprinted Angelman gene. So it's been shut down. So in a regular condition, you have here a normal chromosome from your father, normal chromosome from your mother. You have the gene product from your paternal one that is necessary so you don't have the Prader-Willi syndrome. You have the gene product from your mother, your maternally inherited chromosome, that has the active Angelman gene, and so you're producing both of those products. They're just coming from the different chromosomes, right? One's coming from the paternally inherited one, one's coming from the maternally inherited. On the maternally inherited one, so that you don't have a double dose of these, you then have this epigenetics going on, right? You have the imprinting here, so you deactivate that gene here, you deactivate the gene here. So in a normal circumstance, everything's fine. We're producing all of our normal products, right? But what happens is if you have that condition where there's the deletion, right? If the deletion happens in the maternally inherited one, and so you no longer have these genes, right? You're not producing these genes, and so in your maternally one, in your paternally inherited one, you have a regular Prader-Willi product, right? But now you're imprinted on the Angelman, and because it's the maternally one, you now develop Angelman syndrome, which is a syndrome that is very different. What was the Angelman syndrome? What do they call it? The laughing puppy. Remember, this is the laughing puppy one. The Prader-Willi, which one was this? Does anyone remember? The kids swimming, the ones that just keep eating, right? That was the Prader-Willi. And so very, very different phenotypes, depending on which of the chromosomes doesn't have the deletion. So here, the opposite is happening, right? The deletion happens. Your maternally one is good. If you look up here, your maternally one, you have a regular Angelman gene, right? So you don't develop the happy puppy syndrome, but you have the imprinted Prader-Willi gene. Therefore, it's imprinted, it's turned off. You then develop that Prader-Willi syndrome. That's how they were able to explain same deletion, exact same genes, right? But the end result was dramatically different. You know, obviously, if you were the researcher at the time, you'd be like, you know, pretty confused about that. This began the explanation of it by figuring out what's going on in these situations. We then began to understand the importance of epigenetics. Okay, so, and then these are just sort of DNA methylation of the cysteine. Here's a methylation, here's a methylation, just kind of showing you graphically that, and then here are both of them, the methylation down here on the DNA, the histone modification here, which kind of opens up and upticks the transcription. Okay, so again, go look in the book. There's that 5 pages on. It's more than just kind of epigenetics. It's just basically sort of this gene regulation. It's not a lot of detail. You got probably just as much detail in the book. I mean, in the video, and then also in that paper. If you read this, if you read that paper that I put on Canvas, and you took notes on this, you should be fine. You'll be plenty good for the level of understanding of epigenetics that I want you to have for the exam. Okay, any questions, Pat?
So those are the pages for the book. It's not like the whole book.
No, this is the 6th edition. So if you have the 6th edition, these are. I haven't looked. I didn't even look to see what's in the 5th edition. I don't have a 5th edition anymore. I don't know what happened to mine. So yeah, so those are the 6th edition pages. But then also the paper I'll post on Canvas. It'll say Epigenetics by. It's the author's name. I don't remember it. It's a female. I don't remember what her name is. And then the video. Okay, so now the last sort of molecular genomics thing that I want to just touch on is microarrays. So what I mentioned before is that yes, you know, sequencing DNA and everything was great, but now we are moving on to an area that is even much more complicated, and but with that complexity, it's also we're being able to figure out a lot about epigenetics things. We're being able to figure out a lot about how genes are being turned on and off, and what genes are being turned on and off, and how that epigenome is actually influencing as much of our phenotype maybe as our underlying foundational genome of our foundational genome. Okay, so genes can be regulated at a whole bunch of different levels. This is kind of dark, sorry. This says transcription, post-transcription. This is what we're talking about when we are talking about the transcriptome. Okay, so you have the genome. We mentioned earlier in the semester a little bit about the transcriptome. Basically, the transcriptome is referring to all the genes that are being transcribed, right? So, as you can imagine, you start as a single fertilized zygote, single fertilized egg, right, a zygote, and through mitosis, right, you're going through mitosis, you know, millions, billions of rounds of mitosis. From that single cell, you develop into a fully functional adult organism. All of those, so as you can imagine, you have lots of cells that are very, very different, right? Where does that difference come from? It comes from what genes are being turned on and off. And so we're now at the level of we have sort of the molecular prowess now to be able to go in and kind of figure out in a much finer detail than we ever had before exactly what's going on. And that's just the transcriptome. So now, for instance, I had a collaborator that I collaborate with. He's in Porto, Portugal, and we've done a lot of stuff. That's why I went, I don't know if we talked about the herping trips in Morocco and stuff with him. We used to do all this sequence, we'd go out, and we just basically take samples and sequence it, et cetera, et cetera. And he's been very successful in getting grants and stuff. He emailed me just the other day, and we're going to co-advise a PhD student with him, hopefully, but in Europe, PhD students have to apply for their own grants, and so the PhD students get their own grant. In order to do that, he's like, no, I think in order to get one, we're going to have to do transcriptomes. So now it's moved beyond that. We can't do our old bread and butter, just like easy. We were doing parasite sequencing, all sorts of stuff. This is now the new level. Now with granting agencies, they're all looking for, okay, are you doing the transcriptome? Are you sequencing the mRNA, or are you doing microarrays? That's what I just finished a sabbatical in the Golan Heights in Israel. Did I already talk about that at all? Yeah, that's what we're doing there. And so now, this is kind of where it's all going. So if any of you are interested in doing wildlife research, any research, you're going to have to know sequencing, you're going to have to know that DNA stuff, and it looks like you're probably going to have to start to know how to do these microarrays and things like that. So I want to give you just a little taste or a little background of what microarrays are, even just for people who maybe will never do it, but maybe are going to do interviews, and you can somehow, in your med school interview, throw that in there that you know what a microarray is. Okay, so here's the transcriptome. It's basically your whole set of mRNAs that are being transcribed, right? Which genes are being transcribed and at what sort of density. So we can look at these things and we can look at them in qualitatively which genes are being turned on, and quantitatively which genes are really being turned on and which genes aren't being turned on that much, right? And so that's what we talk about when we talk about the transcriptome. We don't want to confuse that with other post-transcriptional stuff with translation, right? You can also regulate which of your mRNAs are being translated and things like that. When we talk about transcriptome, we're not necessarily talking about that post-transcription translational stuff, right? It's post-transcriptional, but not the translational stuff. Okay, so that's what we mean when we talk about transcriptomes. So in the last few years, it's become possible to look at the entire transcriptome within a single experiment, right? So we could go through, for instance, and say, all right, I want to know what genes are being turned on. Let's say you were in Dr. Marr's developmental biology class, and you say, okay, I want to see what genes are being turned on in Drosophila at the 16-cell stage in this part of the embryo. You could go in there, you could get that, you could look at those cells, and you could say, okay, this is being turned on now. You could then go a week later and say, okay, which genes now are being turned off and which genes are being turned on. There's a lot of variation, obviously, or there's a lot of different combinations of genes that can be turned on and off. But this allows us to understand better lots and lots and lots of different disciplines, right? How exactly are we going from a fertilized single cell to an organism with billions of cells, which is a massive array of different kinds of cells? Okay, so essentially, there are two basic ways of doing this. There's a sequence-based method, and then there's microarrays. We're going to talk about microarrays. We'll kind of mention the sequence-based method a little bit. This used to be really, really expensive, but now with the pyrosequencing that we talked about last time, with that next-generation sequencing, we're now being able to even do this a lot more. That's what we're talking about here. So the next-generation sequencing, this is the RNA Seq. We'll talk about that here in a second. But mostly I just kind of want you to understand what's happening with microarrays, okay? Any questions? Okay, so the genomic analysis of gene expression. So what we're actually doing is we're taking a snapshot, right? So we're taking a cell, right? And let's go back to this developing embryo. We take a cell or take a bunch of cells in a developing embryo, a certain region of the embryo, and we're going to take a snapshot. And we're going to say, okay, what are the mRNAs that are being produced? What are the genes that are being turned on at this specific point in time? Okay, that allows us to then, in a temporal sense, monitor the genes that are being turned on and off. And then, once we know what genes are being turned on and off, we can also see the phenotypic results of those genes being turned on and off, and what's that also going to tell us maybe? It's going to tell us what genes are important for certain things, right? It's good. All along that way, we're going to understand if all of a sudden you are looking at an embryo and all of a sudden a certain gene that you had no idea had anything to do, say it's an eye, this is just totally made up, but let's say it's an eye, some sort of eye pigment protein or something. All of a sudden it's just on fire and it's being transcribed like crazy at this certain stage. That's going to say, oh, wait a second, maybe there's some other function for it. So we're going to be able to learn what the functions of the genes are by being able to see which ones are being turned on and off. Also, and this is a really important one, you can do diagnostics with it, especially with, as you can imagine, cancer. So what's going on with cancer? Cancer is essentially a condition, a disease, where our cell mitosis, our cell replication is going haywire. Therefore, there's probably something that's happening with the regulation of our genes. And so if we go through and we look at, okay, which genes are being turned on, which genes have been silenced, compare a cancer cell, is that the chair again? Compare the cancer cell with a normal cell, and that's going to tell us a lot. That's going to go a long way in figuring out what's going on with lots of different kinds of cancer. Okay, so again, through the cell cycle, provides a whole bunch of data, and it can help us to start to understand how systems as a whole function. So it's some really powerful. Okay, so RNA sequencing, basically, or RNA-seq, basically what we're doing, and there's some overlap with what's going on with microarrays, is we're going to take a cell and we're going to isolate all the mRNA. Once we've isolated mRNA, we're going to convert that to complementary DNA. So basically you have an RNA strand, you go through how are we going to do that, what methods are we going to use probably to do that? Yeah, we're going to reverse transcriptase, a PCR. Basically, we're going to thermal cycle. We're going to make a whole bunch of copies of that, right? So we're going to make a whole bunch of copies of it. We're going to make what we call a cDNA library. That's going to tell us, okay, these are all the mRNAs that are present in what we've isolated from the cell. We're then going to sequence. So now we've done the PCR reaction. So there's an overlap, right, between what we're learning about sequencing and now what we're learning about doing microarrays and things like that. So we're going to sequence the cDNA again with those different methods now with the pyro sequencing and all of that stuff. We can do it in much more efficiently. But just think about the sequencing reaction. You now have a whole bunch of copies of these genes, the DNA that came from the mRNA, right? And then what you do with this is, because you can imagine in a cell all of the different mRNA that is present in a cell or in a tissue or something like that. You're going to be sequencing a whole bunch of genes, right? A whole bunch of cDNAs. You then are going to use that and you're going to just basically blast that against the genome and then figure out what genes are happening. So this is the generation of a lot of data. This takes a lot of money to do, but now as we have developed more technology, this is becoming more and more feasible. Importantly, it gives you a comprehensive and quantitative view. It tells you which genes are being, the more mRNAs they have, the more that it's going to show up, right? The larger the number of mRNAs, that gives you the quantitative, and then the comprehensive is just going to tell you which ones are present, which genes are being sequenced, which aren't. Okay, that's all we're going to say about the RNA sequencing. Okay, so microarrays. What exactly are microarrays? Because of the next generation sequencing and stuff, microarrays we've been doing for a fair number of years, and they've been very, very helpful. It may eventually be replaced by the sequence-based methods, but for now, it's the most feasible one. It's the most likely one that you'll probably do if you want to figure that stuff out. So what we do is we basically create probes for a specific gene, and then we generate complementary cDNA based on the mRNA, and we label them. We then expose those probes to the targets, and it gives us a signal. Okay, that's how we're doing it. Okay, the probes that we make, we affix them, or we sort of lay them down on a couple of different things. Don't worry too much about this, glass slides or nylon or whatever, or even these chips. We now have these microarray chips that we generate. We use a lot of times, we set them down with like ink or printers, kind of. You're just shooting a whole bunch of DNA onto this little tiny area, and then it's affixing to the nylon or to the glass, and then we're exposing it. That may not make too much sense right now. It will here in a second. Okay, so this is the basic dynamics, or the basic steps of what you do when you make a microarray. So you want to attach DNA to some sort of solid support, whether it's glass, plastic, nylon, whatever, right? In each of these little wells, you're going to have a specific gene. So let's say you know a gene. Let's say you want to look at human blood type, I don't know, something, hemoglobin or something, I don't know, whatever. Or let's just do Drosophila. So here you want to look at the vestigial, the gene that codes for the normal wild type wing versus vestigial wing. Here it could be ebony, here it could be et cetera, all of these different genes. You go through, you create a whole bunch of copies of that gene. How do you do that? What method? PCR, right? You then have this whole population of a bunch of genes, or a bunch of copies of a gene, and then you affix it into that little well. That then serves as a probe. Okay, then you do another gene, you do it here. You do it here, here, here. Here's 20 genes here. Here's 20 rows here. So now you have 400 different genes that you can look at. Okay, if you have a whole bunch of these microarrays, you know, eventually you could have, what, 20,000, right? If you knew the 20,000, all of the potential genes, 20 to 25,000 in the human genome. You have all of those fixed to there. You then you then isolate the mRNA from a cell. So let's say we're doing a normal cell versus a cancer cell. Once you have that mRNA, you go through and you make a whole bunch of copies. You make the cDNA, right, to that mRNA, and you label it. That's the important step. It's labeling it. You then expose those two, and depending on which of the probes is affixing to the target, they're going to result in different colors. Okay, that's basically what's happening. And so we'll go through those steps. These are the steps that it shows you in the book. One, you prepare the microarray by spotting the gene-specific genes, right, the oligonucleotides, to each of these different wells, right? So here, gene probe 1, 2, 3, 4, 5, et cetera, and you affix it to this microarray tray or chip. Okay, we do that. Next thing, you isolate mRNA. Here, you're isolating out your mRNA. This is our control. This is a normal cell. This is the experimental cell. Maybe this is the cancer cell, right? You get all the mRNAs from the cancer cell. You go through PCR. During the PCR, you create these cDNAs, but you also label them with a green fluorescence and a red fluorescence. So now you know that the cDNAs that are going to fluoresce green come from your normal cell. The probes that are going to fluoresce red are coming from our cancer cells. So we really want to figure out, okay, what in the cancer cells, what genes have they either turned off, or what genes have they turned on? And so with that MDA gal, right, she had a whole bunch of genes that had been turned off, right? And so then you can develop a protocol where you can try to turn them back on, or vice versa. So this has a lot of potential for the health profession. Okay, so here you have a whole bunch of probes that are labeled either green or red. Expose them to each of those wells. And so here we have this first one in gene 1, it fluoresces yellow. What does that mean? That means, what's that? She said it. What does it say?
Yeah, both.
Both, right? Red and green, mix those together, you get the yellow, right? And so so here that means that both of those are being expressed. So is there any change between the cancer cell and your normal cell here? No, they're both, whatever genes are turned on, they're both being turned on, and they're both being turned on at about the same level, right? So then you go here to the red one. So you have red, what is that telling you in this gene right here? What's happening in the cancer cell? Remember the cancer was red, the normal was green, right? Cancer cell now has something that's turned on that was not turned on in your normal cell. So all of a sudden here you're looking for red and you're like, oh wow, look at this red, 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 red. Look at all of these different genes that the cancer cell has turned on that your normal cell has not. If you have green, so what is this green? What does the green then mean? Your normal cells has that gene turned on, and what's happening in the cancer cell? The cancer cell's turning it off. And so the ones that you're really looking for, right, are those two, the green and the yellow, because that's telling you the change or the difference between the regular cell and the cancer cell. The black one here, what's that telling you? That gene isn't turned on in either of those cells. And so now from here, then you're going to go and you're going to pick out these different ones. You're going to say, okay, what exactly is going on between the cancer and the cancer cell and the normal cell. So that's why microarrays are so sort of powerful. That's why they're so sort of sexy right now, is because it can tell you a lot of really important things. So for wildlife people, there are other things that you can do to try to figure out what's going on. Okay, here, this is a real one. This is an untreated cancer. Oh no, so this is also not just what's going on in cancer cells, but what is happening to a treated cancer cell. So let's say you have a particular drug and you're experimenting with it, you're trying to fight the cancer with it, and in a way you're trying to see how well your medicine either turns on or turns off the drugs, right? So you can have here an untreated cancer cell, here a treated cancer cell, and you can gauge how effectively you are regulating the turning on and off of those genes and things like that. And so there's a whole bunch of different stuff. This picture, not a very good picture, this just shows you those sort of gene chips, the size of them. So they're little tiny ones. You have here, these are the probes, and it just kind of shows you that. Okay, so for wildlife people, when I was doing that research in the Golan Heights, Northern Galilee area, we were looking at salamanders, okay? There were salamanders there. What did we talk about before? Did we talk about the cannibalistic ones, or what did we talk about? Anybody remember? Carl, you remember? So the main part of the project, though, was doing this, was doing microarrays, because there are a bunch of different species of fire salamanders, right? There's a fire salamander in Europe, and there's a fire salamander in the Near East or in Israel, right? Both of them have larval stages where some of the larval stages where they can be either in a stream or in a pond. And then when they emerge from the stream or the pond, they become adults, and all of the adults look kind of the same, right? There's one species there's a species in Germany that does it, there's a species in Israel that does it. So in our project, so we wanted to know, right, there's a dramatic difference between the larvae, right? They're not larvae. Immatures, right? Here, this is actually one here, ambystoma here in the US, but it's kind of similar, right? So here you have a pond form of a salamander, here you have a stream form. As you can see, the pond form, if you look at the gills, they're very different morphologically, right? If you look at just the general body shape, it's different. The coxal grooves here are more pronounced in the pond form. The tail here in the pond form is bigger and wider. In the stream form, it's not so much, it's more narrow. When these emerge as adults, they emerge and morphologically they look much more similar. So what's going on? What's happening in the pond forms versus the stream forms? We'd be interested in knowing how is the organism doing it? How are we going to figure out what's happening? What would you do?
Genes are turned on.
Yeah, so you go through, you would take these salamanders and you would go and you create microarrays and you say, okay, because what has to be happening is through that development during their aquatic stage, right, comparing this one with this one, there's going to be a lot of genes that are turned And then when you get the adults, you're going to say, okay, now let's look at these. Let's see if now, if all of these morphological differences are just based simply on what genes are being turned on and off, could you potentially not have any genes that are different? What do you think? Maybe, right? Maybe it's just completely environmental. Maybe it has to do with, the stream forms don't get much food, and so they can't really develop very good gills. They can't really develop rod tails, et cetera, et cetera. And so microarrays and that kind of technology is even, getting into wildlife stuff, is even getting into, it could be getting, how many of you want to do like zoo stuff? So if you want to work for the zoo, is that true, Corey? Well, so I don't know if I believe it, but if you are having, say there's certain conditions in a artificial setting where your animals are molting or their hair is falling off or something, right? You could potentially use microarrays for that. You can say, okay, what's going on in here? Are the genes, is there something in the cages, is there something in their activity that is turning off or on certain genes? You could go look at microarrays, right? You could say, okay, compare that to a wild one and say, oh, they're not producing this certain protein now because for some reason in captivity they can't produce it. That's why they're doing that. Or that's why they're behaving differently. We see a lot of behavioral differences in animals in captivity versus in the wild. And so really the sequencing the transcriptomes and really understanding the gene regulation is something that's going to go not just in, not just going to, you're not just going to see it in the medical field, but you're going to see it in all these different fields. So that's why I want you to understand it. All right, so understand epigenetics, understand microarrays, and that's it.